Hi, my name is Lisa Lang, and I am a scientist with QIAGEN. Today, I would like to introduce you to the Human Somatic Mutation Database, Hsmd. The scientific literature describing genetic alterations and associations relevant to translational research is large and growing rapidly. Searching through the scientific literature and digging through multiple websites to find the relevant information can be daunting and takes a tremendous amount of valuable and irreplaceable time. Prior to joining QIAGEN, I was a variant scientist for over 5 years, and I would often feel like I was drowning in a tsunami of information. Some online databases attempt to house content, but unfortunately, the content is outdated or incomplete. So what if you could access a database that would allow you to find the answers that you're looking for faster? The Human Somatic Mutation Database, Hsmd, is a web-based application containing extensive, expertly curated content relevant to somatic cancers. Hsmd includes over 300,000 publications with key findings from the literature summarized by our team of 100 PhD scientists. Hsmd also provides valuable insights on genes and alterations that have been previously observed in over 250,000 clinical oncology cases. Users can explore at the gene, alteration, disease, drug, or trial level to get to that key finding that they're searching for. Hsmd allows users to easily search the largest, most up-to-date somatic knowledge base, quickly and accurately determining the biological relevance of somatic alterations. It is indeed the fastest growing knowledge base with more than 5,000 manually curated alterations added each month. Clinical trials, drug label information, and information from the primary literature are updated on a weekly basis, with guidelines and content from the integrated databases updated quarterly. This database enables researchers and scientists to explore whether a gene is a tumor suppressor or an oncogene, the number of associated variants, disease treatments and trials, and also to get a sense for the impact of associated variants and whether these variants are pathogenic variants or whether they're tier 1 or tier 2 variants, for instance, based on the guidelines from ACMG and the ASCO-CAP guidelines, respectively. Somatic frequency information is provided across somatic cancers at the gene and variant level so that you can readily get a sense for if this variant is rare or if this variant is seen most readily or often in diseases such as non-small cell lung cancer or colorectal carcinoma, which could be very helpful to helping you to design your experiments. The actionability information, as mentioned, will bucket the variants in the tiers that are associated with those guidelines. And that will also consider the coding impact. So you can really look across the nonsense alterations or frame shifts and determine what number of these variants have been categorized as a particular tier, meaning that there are actually variants of unknown significance, which might be good targets for further explorations. And clicking on the links will actually take you to a list of all of those variants. We also do include a distribution chart so that researchers can immediately determine if an alteration resides in a seemingly hotspot region. Using the functionality to the right, you're also able to filter and sort based on variants that are known to be activating or inactivating alterations. And you can also change the isoform to your preferred transcript. Hsmd does provide gene and variant level comments written by PhD scientists, and these comments will all have the references very clearly indicated so that you can go to that source information. So transparency is very important in terms of how we present this data to you. As you can see here, the alteration details section will show you the specific variant impact for various transcripts of interest. And for the treatment trials and literature, our scientists will also go ahead and read all of the key findings from, for instance, the drug label and pull out that information or from the publication. And also will include any clinical trials so you can get a sense for what are the other inclusion criteria or exclusion criteria for trials that are currently recruited. And of course, the literature finding, which again, we are updating on a weekly basis. So this really, this up-to-date content really will allow you to ensure that you reduce the risk of missing any important findings. And in essence, Hsmd is going to allow you to reduce the time that you're taking to do this kind of research by allowing you to quickly explore across cancer types or drugs and other attributes, and also search in depth information on variants or genes in one place. Whether you're trying to determine if a variant you identified is indeed novel, educate yourself on what is published about a specific gene as you work on your manuscript, or design an experiment using that perfect experimental design, Hsmd is going to allow you to know more in less time and with less effort. Thank you so much for your time and attention today.